One of the reasons why I've really enjoyed working on fruit flies is that what you learn in the fruit fly can have immediate applications to insects that are tremendously important in the real world.
John's changed medicine in two ways. One is just working out the fundamental biology of how we smell. The other is I just think it's going to have impact on human health.
He's zeroed in on chemoreception in insects. Chemoreception in insects has so many different facets. It's important for crops, for agriculture.
In the insect world, it has a lot of implications in terms of vector control for things like mosquitoes, either to repel it or to attract it to its doom, or to control its mating habits in order to control the vector for malaria.
I grew up in Ames, Iowa, which is a quiet university town, a very nice place to grow up. And when I was young, I was very interested in the diversity of animal life. And I was particularly interested in marine biology. After I graduated from high school, I got a summer job at the Marine Biological Lab at Woods Hole, Mass. And the job was to deliver big carboys of distilled water, which were very heavy. But then I used to look around the lab and ask people what they were doing. And some of them let me participate in experiments, which was really exciting for me. It was my first real exposure to research. My interests were actually pretty broad when I started at Harvard. I was interested in a lot of different things. But the spring of my freshman year, I took an introductory biology course that just was tremendously exciting to me. So at the end of that year, I declared a major in biochemistry. So I applied to Stanford, the Stanford Biochemistry Department, and I flew out for an interview. And I remember thinking to myself that I'm not in Massachusetts anymore. Then the next day, I had interviews with members of the faculty. The interview I remember best was actually the one with Paul Berg and Dale Kaiser in Dale's tiny office. That and some other interviews that went well really convinced me that this would be a tremendous place to be a graduate student.
So the biochemistry department back then was an incredibly vibrant place. You had basically all these giants in their field. Recombinant DNA was invented before that and was spilling over, and all the miraculous things you could do with this was just incredible.
In the department at the time, there was an ethos of both rigor and of courage. And the rigor, I think, came straight down from Arthur Kornberg. One was expected to have a very high standard of thinking, and of the quality of one's data had to be really high. The other really critical component of the ethos in the department, I think, was courage. People were working on very ambitious projects. People were taking great risks in order to basically solve great problems. I arrived at the Hogness Lab at a very exciting time. Dave had conceived the notion of a genomic library. Science can be a bit like an ice skating rink sometimes, and there are a lot of hockey players on that rink, but Dave, by contrast, was a figure skater. His science was very elegant.
When I first met John, actually, he had started his postdoc a little bit before me, and he told me what he was doing, and it was this incredibly wild idea, and this, I think, typifies John. He actually was going to express antibody genes in yeast to be able to try and inactivate genes.
So I needed a lab which knew how to make monoclonal antibodies, which was fairly early back then. And someone suggested Irv Weissman in the pathology department at Stanford, and actually managed to raise monoclonal antibodies against a yeast enzyme and getting yeast to make an antibody which would actually bind to this protein and neutralize it. And I found evidence for an effect in vivo too.
It's one of these far-fetched things that Stanford's kind of known for, and quite frankly that John's known for, something really original and creative.
I think part of the reason why Mike and I were so close was because we both had been instilled with the same values that we picked up in the Stanford Biochemistry Department. I'm very grateful to Stanford for having encouraged me and given me the willingness to try something new and different, and take risks.
When he got to Yale, he looked around the field, or he looked around the world of neurobiology, and he said to himself, I think, what needs to be addressed? What is the problem, and how can I address and solve this particular problem?
So we developed an in vivo expression system which uses a mutant fruit fly in which one particular nerve cell in the antenna of the fruit fly has no receptor. Using technology that was originally invented by Paul Berg, we were able to construct genetically engineered fruit flies in which this one neuron, which has no receptor of its own, now expresses a receptor of our choosing.
His labs use bioinformatics very effectively. This was one of the great tools that allow them to have this breakthrough of identifying odorant receptors in Drosophila. He's a superb geneticist, so he has all that. And I think because John was at Stanford Biochemistry, he also really appreciates the power of biochemistry.
He was the first one to recognize the molecular organization of odor receptors in Drosophila. He was the first one to begin to dissect the molecular mechanisms, the regulatory mechanisms that govern the choice of which receptor, gustatory or odor receptor, is expressed in any one neuron. These are landmark achievements.
The system that he developed genetically has been used by a lot of labs in the Drosophila and the insect world and mosquito world as well to carry out these really important basic characterizations of odorant receptors. And I think that's had a huge impact in the field.
Every year, hundreds of millions of people are infected by diseases that are transmitted by insects, diseases like malaria, dengue fever, yellow fever. And many of these insect vectors that transmit these diseases find us through their sense of smell. So if you can understand the molecular mechanisms that operate in these insect vectors of disease, we may be able to come up with new ways of preventing them from finding humans, or trapping them in traps, or repelling them, or confusing them.
Insect control is, huge in human pathogenesis, so we've really got this under control, and I think his biology is going to be fundamental for that.
The number of students that are committed to going into John's lab and interested in pursuing their PhD degree with him is extraordinary. I see them later at chemical senses meetings, for example, and they continue to talk about the influence that John had on their career.
He's not just a good teacher, he's a phenomenal teacher. He personalizes his lectures, and it just resonates really, really well with students. I've taken a lot of my own lessons from watching John teach.
So I started teaching a course to non-scientists where we explain basic principles of biology, but in a way that's designed to be interesting to them. We place it in the context of social issues like the AIDS epidemic, for example. And I think they emerge with a very healthy respect for the importance of evidence and reason in making decisions.
He's gone out from Stanford, having acquired a fabulous education and motivation and excitement, and he's taken that and passed it on to a whole new generation of students who, in turn, are passing it on to their students.
He embodies the style and the quality of science that I think any institution would love to showcase, number one. Number two, he is a wonderful mentor.
I think he needs to be recognized not only for his research abilities and his research accomplishments and his accomplishments as an educator, but also as an ambassador of Stanford.
It's really a wonderful feeling to receive an award from the people who gave you your education, who evidently think you've done something special with it. And it's especially wonderful to receive an award named after Arthur Kornberg and Paul Berg, for whom I have such enormous respect.
I think John's an incredible choice for the Kornberg-Berg Award because he's done field-changing science. I think he's brilliant, inspirational, and he's just getting started. I'm sure we have a few decades ahead left of original research out of John.